Hello everyone. Today we are going to study microbiology. Our topic is sterilization and disinfection. We are going to describe it in details. First, let's see sterilization. What is the difference between sterilization and disinfection? The difference is simple. In sterilization, substances is made free from all microorganisms both in vegetative and sporing state, whereas in disinfection, spores are not affected. Sterilization is further of two types: physical method and chemical method. Physical method sterilization again consists of sunlight, drying, heat, dry and moist, filtration and radiation. Whereas chemical methods consist of alcohol, like you have seen ethyl alcohol, isopropyl alcohol are such examples, aldehyde like glutaraldehyde, formaldehyde, dyes, halogens, phenols, surface active agents, metallic salts and gases. Today we will focus on physical method of sterilization, and in coming session we will see chemical method of sterilization. I have made the slide slower so that you can note down the points. Now the most reliable method in physical method of sterilization is heat, and it should be method of choice. Now there is a term which is ever asked in exam, TDT, that is thermal death time, which is the minimum time required to kill a suspension of organism at a predetermined temperature in a specified environment. The remembering point here is TDT, thermal death time, is inversely proportional to temperature. That is, if TDT is increased, temperature is decreased. And you must remember this point. Now let's elaborate heat. As we already mentioned, heat is of two types, dry heat and moist heat. And it is further subdivided, like dry heat in flaming, incineration, hot air oven, and moist heat in temperature below 100 degrees Celsius, temperature at 100 degrees Celsius and temperature above 100 degrees Celsius. Both in three groups, right? Like dry heat in three groups and moist heat in three groups. Now let's individually know each term, like flaming. What do you mean by flaming? It's simple, heating over fire till they become red hot. Incineration in simple words simply means burning. Burning of organic substances contained in waste material. Now the most important point asked in exam is hot air oven. Mostly come as short notes. It's an electrical device. Temperature range 50 to 300 degrees Celsius with thermostat controlling temperature, and most widely used method. Now how you will test if the hot air oven is working or not? For knowing this, we use Clostridium tetani spores, Geobacillus and Brownie's tube, and also strips. Strips are placed in the hot air oven. Oven is switched on and the strip changes its colors, which shows working condition. Now let's see moist heat. As we know, it is categorized into three groups: temperature below 100 degrees Celsius, temperature at 100 degrees Celsius and temperature above 100 degrees Celsius. Now what's come under temperature below 100 degrees Celsius? You all have heard about pasteurization of milk. In simple terms, it's sterilization of milk. Important point, its holding period is 63 degrees Celsius, 30-minute holder method, or 72 degrees Celsius, 15 to 20 minutes, followed by cooling quickly to 13 degrees Celsius or lower. Temperature below 100 degrees Celsius target all non-sporing pathogens. Examples are Mycobacteria, Salmonella, Brucella, relatively heat resistant, Coxella burnetii. Now temperature at 100 degrees Celsius. Boiling. but it's not recommended for sterilization. And sterilization may be promoted at 2% sodium bicarbonate. Next is steam at atmospheric pressure 100 degrees Celsius. It's an inexpensive method used to sterilize culture media. Now important part is autoclave. Autoclave device that use steam to sterilize equipments and other objects. All bacteria, virus, fungi and spores are inactivated. Holding period varies. Temperature 108 degrees Celsius, 147 degrees Celsius. In between them, items, dressings, instrument, labware, media, pharmaceuticals, product, and the recommended temperature and durations. Temperature is 121, then duration is 15. Temperature is 126, duration is 10, and 134, then duration is 3. Hot air oven and autoclave come as short notes, so you must jot down the important points. Next comes filtration. Filtration, it's candle filters like used to filter waters. Then you must know about radiation, how UV radiation we use it as sterilization. It's used in laminar airflow for 20 minutes. So now we have finished our topic on sterilization, physical method of sterilization. Next we will discuss on chemical method of sterilization. Till then, take care.